This is the last video for the Mendelian Inheritance Unit. In this video, we will discuss human genetics and the role of using human pedigrees to study inheritance of traits. Seeing the segregation of alleles in humans is not as easily observed as in pea plants, because humans do not make good study subjects. You would have a difficult time trying to choose two humans to mate so that you can study what traits are observed in their offspring. Additionally, the number of children in human families is relatively small, so Mendelian ratios are much harder to observe because of random fluctuations due to chance alone. For example, although you have a 50-50 chance of having a male or female child, there are some families that have all daughters or all sons. Because we cannot use controlled matings, instead we use human pedigrees or family trees. When you examine a human pedigree, the symbols have specific meanings. All the circles are females and all the squares are males. An open symbol means that the person does not have the trait you are studying, while a filled-in symbol means that they do. Horizontal lines between the shapes represent matings, while vertical lines lead to the offspring. The siblings in a generation are connected with a horizontal line above the symbols and are arranged from left to right in the order of birth. Other symbols which are not seen as often include a double line indicating mating between relatives and lines identifying twins. When studying human genetics, you will use pedigrees to determine if the trait you are studying is inherited in a dominant or a recessive manner. In other words, is the trait controlled by the dominant allele or the recessive allele of a gene? This pedigree shows what it looks like in a dominant pedigree. It is tracing brachydactyly, which is a rare trait in which the middle bone of the fingers fails to grow, leading to shortened fingers. This pedigree suggests brachydactyly is controlled by a dominant allele because most matings that produce offspring have one affected parent, and typically about half the offspring are affected. A key to look for in a dominant pedigree is that if there is an affected offspring, there must also be an affected parent. Additionally, the trait appears in most generations, although this is not a requirement. In contrast to a dominant pedigree, a recessive pedigree has the following characteristics. Individuals who are affected may have unaffected parents, and the trait may often skip one or more generations, although this is not an absolute requirement. The pedigree shown is for a family affected by albinism, a condition that results in reduced amount of melanin pigment in the skin, hair, and eyes. Once you have determined if your pedigree is recessive or dominant, you can use that information to predict individual genotypes. In this pedigree, albinism is a recessive trait, so anyone who is an albino, indicated by the shaded shapes, must be homozygous recessive. Each of these shaded individuals would have the genotype little a, little a. From there, you can often fill in other information. For example, these two parents must be heterozygous because they do not show the trait, but they had to have the recessive allele to pass to the next generation. They were both carriers of albinism. You will not be able to determine every genotype, however. For example, these sons could be homozygous dominant or heterozygous based on the parents. You are unable to know which it is. Although you do not know which genotype is for sure, you can develop probabilities. These sons would have a 1/3 chance of being homozygous dominant or a 2/3 chance of being heterozygous. You should draw a Punnett square and examine the fertilized zygotes to determine where those probabilities came from. Many traits with single gene inheritance demonstrate complications that can obscure the expected patterns in pedigrees. Certain traits show incomplete penetrance, in which not every individual with a genotype displays the corresponding phenotype, either because of environmental effects or because of interactions with other genes. This is seen here, where each circle represents an individual with the same genotype. However, the purple color phenotype is only seen in 5 out of the 10 individuals. This trait would be said to have a 50% penetrance. Another complication is when traits show variable expressivity. This is when a phenotype is expressed with different degrees of severity in different individuals. The trait is always expressed, but it is seen as different levels. We can see that here, where all 10 individuals are showing purple color, but the darkness of the purple is variable. In humans, there are a variety of traits that we have identified as being inherited with standard Mendelian genetics, such as freckles or widow's peak. There are also a number of disorders which are inherited following Mendelian principles. These are considered genetic diseases because the disorder is caused by a mutation in the DNA which is passed from one generation to the next. The majority of genetic diseases are recessive disorders, although there are a few examples of dominant disorders. In order to remain in the population, a dominant disorder must not affect someone's ability to reproduce because there cannot be an unaffected carrier for dominant disorders, the way there can be for a recessive disease. As we have learned more about the genetic mutations that cause genetic disease, we are now able to perform genetic testing to identify the genotype of an individual in order to diagnose them with one of these diseases. There are now many companies that will carry out genetic testing on adults in order for individuals to learn what diseases they may be a carrier for, or what genotypes may give them a higher risk of disease, such as breast cancer. The benefits of these tests are to provide more personalized medicine and help you manage your risks if you are more likely to develop a disease. Genetic testing can also be carried out in utero to determine if a developing fetus has a genetic disorder, and immediately after birth to identify any genetic disorders that need to be treated from birth. Anyone who was born in a hospital has been tested for a panel of certain genetic diseases for this reason.